Hi everyone, my name is Marie and I will be your host for today. I'm happy to welcome you in today's webinar on Adeno development, solving challenges for vectors that are easy in theory but complex in practice. Before we begin, let's cover a few housekeeping items. First of all, today's session will be recorded, so the replay will be shared and available afterwards. There is also a Q&A box where you can drop any questions at any time that you want during the webinar. And then at the end, during the Q&A session, we will answer as many questions as we can with Katie. And you also see that there is a resources panel in which you can find some resources to learn more about our services and also about our company, SK Pharmteco. Now, I am pleased to introduce our speaker, Kelly Tran, who is the manager for upstream viral vector process development at SK Pharmteco North America. Kelly is a process development leader with over 12 years of experience in biopharmaceuticals, focusing primarily on suspension cell culture development and optimization of bioreactors for several modalities, just like monoclonal antibodies, glycoprotein, and viral vectors such as LVV, Adeno, and AAVs. Before joining SK Pharmteco, Kelly was working on the COVID-19 vaccine and LVV tech transfer at Janssen Pharmaceuticals. Kelly also holds a MS in biotechnology from the Pennsylvania State University. We're thrilled to have Kelly today to share some practical insights on adenovirus process development and the challenges of moving from early process development to scalable manufacturing. Please join me in welcoming Kelly.
Thanks so much, Marie. And hello, everyone, and thank you for joining today. Like she said, I'm Kelly Tran from Viral Vector Process Development here at SK Pharmteco in King of Prussia, Pennsylvania. And I'm going to be talking today about our adenoviral vectors and how we approach scaling and challenges here at SK Pharmteco. We know adenoviral vectors are well characterized and versatile, but scaling them from early development to manufacturing can sometimes be more complex than it appears. A process that performs well at a benchtop scale might become less efficient or less consistent at a larger scale, and adherent platforms can limit the path to scalable production. So in this presentation, I'll share our practical insights from our adenovirus development and manufacturing projects, including how targeted process optimization and scale-up planning can help improve your process robustness, reduce risk, and support predictable transition into manufacturing. And feel free to ask any questions at any time with the Ask a Question button at the top of your screen. So feel free to ask questions at any time. Before we dive into the technical content, I want to briefly introduce SK Pharmteco and our parent company, SK Incorporated. SK Inc. is one of South Korea's largest conglomerates with a large global foundation across several industries, including energy, chemicals, semiconductors, telecommunications, IT, and biopharmaceuticals. SK Pharmteco builds on that stability and investment capacity as a global CDMO, supporting customers from early development through commercialization across viral vectors, small molecules, and peptides as well. For viral vectors, our focus includes AAV, lentiviral vectors, and adenovirus, intending to help clients advance complex therapies with reliable development and manufacturing support. And we're not just focusing on today's process, we're constantly developing next generation platforms, especially in complex areas like adenovirus, to stay ahead of evolving clinical and regulatory needs. And the work here I'll present today reflects that mindset. So SK Pharmteco is a CDMO that has a global network of strategic facilities across North America, Europe, and Asia, designed to help support flexible and resilient supply. Our viral vector manufacturing sites are located in King of Prussia, Pennsylvania, where I'm at, and south of Paris, France. While our broader network also includes a small molecule manufacturing, process R&D, analytical services, and particle engineering capabilities. This global presence helps us support clients across development and manufacturing needs, while also reducing the supply chain risk and providing regional flexibility as programs advance. At SK Pharmteco, our mission is centered around helping clients bring life-impacting medicines to patients around the world. We do this by combining our technical expertise, advanced manufacturing capabilities, and a customer-focused approach to support partners as they develop and deliver these innovative therapies. Our vision is to be the most trusted global partner in delivery of innovative medicine, and that commitment guides how we support programs from development through manufacturing. So this slide gives a quick snapshot of our viral vector manufacturing capabilities. Across our sites in France and the US, we support AAV, lentiviral vector, adenovirus, and other viral vector programs with experience across more than 100 cGMP batches. For adenovirus programs, we provide process development and scalable manufacturing designed to help bridge that gap between early development and performance for reliable cGMP execution, which is the focus of today's webinar. So in today's session, we're going to go through the full life cycle of the adenovirus development here at SK Pharmteco, starting with early stage process development and moving through upstream and downstream manufacturing considerations. We'll walk through how we think about adenovirus manufacturing, starting with our vector design, and then how we build the process, how we scale it, and ultimately how we drive performance. So we'll also discuss critical challenges such as vector yield and process consistency as well. Then finally, we'll examine practical strategies for scaling from research and clinical phases to commercial manufacturing, all while maintaining that product quality and cost effectiveness. At SK, we like to emphasize on designing to scale at the beginning, so we're building for efficiency and engineering for success in the end. So I want to talk about some of the platforms we support first. This slide highlights the evolution of the adenoviral vector design from first-generation systems to helper-dependent adenovirus, or HDAd, which sometimes is referred to as gutless vectors. In the first-generation adenoviral vectors, at the top here, you see the E1 region is deleted to make the vector replication deficient. However, these vectors still retain several viral coding regions, so because of that, there's still some risk of replication-competent adenovirus, or RCA, formation, along with increased immunogenicity and limited payload capacity. Then in the lower schematic, we have the HDAd platform, where essentially all the viral coding regions have been removed, leaving only the ITRs and the packaging signals required for vector propagation. So this creates several important advantages. First, the risk of RCA generation is effectively eliminated. And then second, the payload capacity is significantly expanded to greater than 30 kilobases. I believe it's almost 36, allowing for delivery of larger, more complex genetic constructs. And then third, removing those viral genes helps reduce host immune response and then improves overall safety profile. Overall, the evolution towards simplified vector design enables more flexible therapeutic strategies while supporting improved safety and larger transgene capabilities. And at SK Pharmteco, we support both first generation and HDAd platforms, allowing us to tailor vector design to the specific needs of each program and therapeutic application. Okay, now we can move into our development strategy. This slide describes the upstream process. Development strategy focusing on how production process is optimized and scaled from early development to pilot manufacturing. The development usually begins in shake flasks, which is just used for high-level screening for key variables like cell lines, if applicable, MOI, VCD at infection, things like that. These experiments help to identify more promising biological conditions that can then be used to move into bioreactor studies, where we have more controlled bioreactor parameters and they're evaluated. That includes the optimization of pH, DO, power per volume, product duration, and a feed strategy if necessary. These studies help to confirm the shake flask findings and then also see if they translate well into bioreactor conditions. We sometimes use the Ambridge 50 for more in-depth DOEs, depending on program needs and timeline if they want to optimize more. Or we can go straight into the 2-liter or 10-liter benchtop reactors to help soft lock those parameters. The 10-liter reactors also help to provide material for downstream processing studies. And it also helps to ensure process stability and scalability and reproducibility. So finally, the process is scaled all the way up to pilot production, which we have a 50, 200, and a 500, where the goal is to establish a robust manufacturable process suitable for large-scale production. So overall, this slide outlines a structured stepwise approach to upstream development from early screening to pilot manufacturing, ensuring our process understanding, consistency, and scalability. We focus on making a scalable process from the start so we don't have to re-engineer it later. So this slide lays out the full upstream operations, showing how material moves from initial cell thaw all the way to harvest. In the upstream flow, we start with vial thaw. The cells are expanded stepwise, first in shake flasks, and then depending on final production size, it can be followed by cell culture bags and/or stir tank reactors. Depends on the cell mass required in the final bioreactor scale. We've inoculated pilot reactors from shake flasks, cell culture bags, and SDRs, depending on what you need. The process then moves into production bioreactor where infection occurs, usually with viral seed stock, to help generate that target product. And then at harvest, key additions that are made are lysis buffer with between 20 to break open the cells, and an endonuclease to reduce HCDNA. And then also a salt quench with sodium chloride to help stabilize that material. And then it's handed off to downstream for further processing. Here are some examples of a successful scale-up. The slide highlights one of the key goals in our adenoviral vector manufacturing, which is achieving consistent cell growth and viability as we scale from development all the way to pilot scale without needing to re-engineer the process. So you can see across several graphs here that the progression from small to large scale, as the cells progress and transition through infection and production phases, we see similar trends and performance aligning across all scales. This demonstrates a strong process robustness and reproducibility during scale up. This also indicates that the process parameters, including things like mixing, gas transfer, and infection conditions, are translating effectively across all reactor sizes. And for adenoviral vector, we know that maintaining that cell health and growth kinetics across scales is extremely important because vector productivity is highly dependent on infection timing, metabolic state, and cell viability. So if scale-up introduces major shifts in growth behavior, it often requires re-optimization, which adds time, costs, and regulatory complexity. So what these examples demonstrate is a scalable, well-characterized upstream platform that helps to enable our programs to seamlessly transition from process development into manufacturing. And ultimately, this reduces scale-up risk, accelerates developmental timelines, and supports reliable adenoviral vector manufacturing for clinical and commercial supply. So now that we've looked at consistent cell growth and viability across scales, the next question is whether that translates into consistent adenoviral vector productivity at harvest. So this slide helps demonstrate that the viral titer remains highly comparable as the process scales from development into larger stirred tank reactors. And the key takeaway here is that the process delivers robust and reproducible titers across multiple scales without significant loss in productivity during scale up. So that consistency is essential for reliable manufacturing and predicting clinical supply. And we can see here across 2 liter, 10 liter, 50, 200, and 500 that the overall titers remain tightly aligned with each example indicating a robust and reproducible process. We've also used the AMBER 250 for early development and late phase development here with very comparable productivity. So for adenoviral vector manufacturing, maintaining that viral titer during scale-up is crucial because productivity can be highly sensitive to process conditions like mixing, oxygen transfer, infection timing, and cell physiology. So when those parameters aren't properly controlled during scale transition, you often see variability in vector yield and overall process performance. So what this data demonstrates is a well-characterized and scalable process that minimizes the need for redevelopment or extensive optimization at larger scales. That then can transfer into faster tech transfer, lower manufacturing risks, improved batch-to-batch consistency, and greater confidence when moving from those early development phases to clinical and commercial supply. And overall, these examples show that the platform is capable of maintaining strong adenoviral vector productivity across a broad range of bioreactor scales. which supports a seamless path from process development to manufacturing scale. But sometimes we do face challenges when scaling. This slide highlights a real-world downstream process scale-up challenge that we had during one of our transitions to pilot scale, and how the team successfully optimized that process to maintain both product quality and recovery. So the primary challenge that we observed was we saw product breakthrough during the capture step, along with significant downstream fouling. While the process performed well at smaller scales, we noticed that when scaling to the larger pilot scale, it introduced a higher impurity burden when entering purification. So that impacted the resin performance and overall efficiency. And throughout, the team did a real-time root cause investigation that identified that the impurity to product ratio had increased substantially at the larger scale. So to address this, again, in real time, they did several targeted optimizations during the run. The team redefined the column loading strategy, adjusted the resin selection and operating conditions, and also introduced improvements to the polishing steps to help better manage that impurity clearance. So together, these changes improved the process control and reduced the fouling risk later on for further operations. So the outcome was a successful adaptation of the process to the manufacturing scale without compromising that product quality or critical quality attributes. So this case study demonstrated the importance of a scalable DSP design and being proactive for process optimization when moving from development to larger scale production. So speaking of downstream, let's move on to the downstream optimization. This slide highlights the structured approach to optimizing the downstream process, starting with upfront clarification and then moving through key unit operations. The downstream process flow begins with clarification to remove cell debris and reduce turbidity. Our clarification strategy is based on turbidity from the VCD, specifically whether we need to do a single stage or multi-stage approach is required. Next, moving into TFF, or tangential flow filtration, is evaluated as a conditional step. Depending on your process and your volume titer, you might not need it. So it's, again, conditional. But when it is used, it's optimized for impurity reduction and maximizing yield recovery. Then we move into capture chromatography, which isolates the product of interest. And then with this anion exchange, the focus is selecting the appropriate bind dilute strategy. The team focuses on optimizing buffer conditions like pH, conductivity, and evaluating column options from different vendors. Following that, they have the polishing step to help further improve purity and remove any remaining residuals. So for this polishing step, ion exchange, the aim is to minimize loss of adenovirus while maximizing residual reduction by flow-through process. And again, they use buffer optimization, column selection, and screening different modalities to remove remaining impurities or residuals. And then finally, we have a second TFF, TFF2, is used for final concentration and formulation adjustments. This step is just primarily used for buffer exchange and volume reduction to help prepare for the final drug product. So overall, the theme is stepwise data-driven optimization, where they balance yield, impurity clearance, and process efficiency at each stage. And here we have some examples of that. This slide highlights the effectiveness of the downstream process in reducing the host cell protein while also maintaining product recovery across each unit operation. So the graphic on the left, starting with HCP load at TFF1, we have a high HCP load, then we see a substantial drop during the capture step, followed by continued reduction through polishing stages and final TFF. While there are some fluctuations in concentration due to changes across steps, the overall consistency and total HCP burden is reduced to 20 nanograms per mil, where other processes we've seen 400 to 1500 nanograms per mil. So overall, by the end of the start to finish, it's 99% reduction in HCP. And then on the right, we see viral recovery for each step during the downstream process. So the final recovery from start to finish of the downstream process is 75%, which is higher than the current industry standard of 50 to 60. And overall, that demonstrates a process robust in purification that successfully balances high viral genome retention across multiple complex unit operations. So the key takeaway is that the purification strain is highly effective and robust at achieving significant impurity clearance while maintaining that process consistency and recoveries, even when transitioning between different upstream feeds. So how do we scale up? A successful transition from benchtop to manufacturing scale requires total alignment across processes, equipment, and the facility to maintain a performance at larger scales. So to ensure a transition is smooth, we focus on 3 core pillars. In the upstream scale-up, we need to account for larger volumes, longer processing times, while also implementing a specific strategy designed to preserve that critical mixing and aeration dynamics. For the downstream scale-up, it's crucial to select fit-for-scale filters, columns, and equipment, as any shifts in the upstream performance can directly impact downstream purification efficiency. So, and finally, the operational readiness. So beyond the science, we manage logistical realities of there's increased footprint for larger reactors, larger equipment, more buffers used, and also more waste generated. So ensuring that both the equipment and the facility are designed to support that robust automation as well. So scale-up requires alignment of both processes, equipment, and facility capabilities to scale up successfully. So some key takeaways for a scalable manufacturing of adenovirus. Achieving scalable adenovirus manufacturing requires a strategic shift from early process design to manufacturing ready execution through several key actions. First, we need to optimize early by designing small scale studies to identify those critical parameters that build a strong foundation for our future scale up. It's equally important to plan for scale by transitioning developmental data into specific requirements for equipment, facility layout, and process controls. Throughout, we're always adapting with data and using real-world performance results to troubleshoot, refine, and effectively reduce manufacturing risks. And finally, because the adenovirus production is not one-size-fits-all endeavor, it's necessary to tailor the strategy to reflect each unique process needs, quality goals, and specific manufacturing constraints they might have. So ultimately, a scalable process is enabled through a combination of early planning, deep process understanding, and a target optimization. So why SK Pharmtechco for adenovirus manufacturing? Here at SK Pharmtechco, we provide a comprehensive solution for adenovirus manufacturing by guiding programs from initial process design through full manufacturing readiness. We offer integrated expertise in adenovirus development alongside scalable upstream and downstream processes, designs that ensure technical success as the volume increases. So this is further supported by our global manufacturing network designed to meet the needs of both clinical and commercial supply. And ultimately, the goal is to help adenovirus programs transition from being feasible in an early development setting to becoming reliable and robust in a manufacturing environment. Thank you for joining today. That's all I have, and that concludes this presentation portion of this webinar. I'd be happy to take your questions during the Q&A session. You can also reach out to SK Pharmtechco to learn more about how we can support your adenovirus or any viral vector program requirements. Please visit our webinar, our website, and connect with us on LinkedIn. There's a QR code here that you can go to on the slide. So thank you.
Thank you very much, Kelly, for your presentation. It was great to hear your perspective on adenovirus development and all the practical challenges that can arise from moving from early process work to scalable manufacturing. So we received a few questions from the audience. So we are now going to move into the Q&A portion of today's webinar. And I'll start with one question that tied closely on the early process development discussion. And the first question, Kelly, comes from Christine. And it's based on your experience. What are the most critical process decisions in early adenovirus development that can either enable or jeopardize a successful scale-up later
on? One of the biggest factors, I think, is selecting a cell culture platform, whether you're going from adherent or suspension, and then whether you want to move from adherent to suspension, and what cell line you want to use. Then parameters such as viable cell density, infection, MOI, infection timing, and the metabolic state of the cells is important as well. That impacts vector productivity and consistency during scale up. A process that performs well at 2 liters can become unstable or at 200 liters to 500 liters if those parameters are not well characterized early on. So having a good understanding of that in early development is important. Another critical area is designing a process around scalable engineering principles. Things like mixing strategy, oxygen transfer, shear sensitivity, if applicable, and feeding approach need to translate from across reactors, geometries, and scales. If the process is overly dependent on unique small scale systems, it often requires a lot of engineering later. So just making sure you have those critical parameters studied early on is important at small scale.
Okay, thank you, Kelly. That's indeed a helpful reminder that scale-up success really start much earlier than manufacturing. We received another question that comes from David, and this one is about common pitfalls during scale-up. So when scaling up from benchtop to manufacturing, what are the most common pitfalls that you can see, and how can developers proactively address them before they become major issues?
So some of the common issues we've seen are not thinking about intermediate stability. At benchtop, it can take a few hours to do a process or certain stages of a process, but when you scale that to manufacturing, it can take much longer. So it helps to build in to your early development studies the intermediate hold studies for a realistic approach. If you know certain steps are happening on which days, where you need that stability data. Another issue we've seen is supply chain issues. Long lead times for materials can cause delays, but performing like a raw material assessment early on can help mitigate that, and finding a secondary supplier if you can, just so you don't have to wait on those materials. Also verifying early on that all your materials are CGMP grade. We've had some programs that were using raw materials that only had an R&D research grading available. But luckily for them, the vendor did release a GMP grade material in time for them for manufacturing. But just doing that due diligence for a raw materials assessment upfront can set you up for success.
Okay, thank you, Kelly. So another question that we received from the audience is, what are the key considerations when transitioning from adherent to suspension cell culture for adenovirus production, and how do you ensure product quality remains comparable?
So there's a lot of things to consider when moving from adherent to suspension. One of the things that stands out is impurity profiles. The suspension cell cultures can get much higher cell density at infection, so that can lead to a higher impurity profile from the residual host cell, so HTDNA, HTP. Just making sure your downstream process can clear that is good, just to reduce it to regulatory guidelines. Another thing that comes to mind is the cell line itself. If you're adapting your adherent cell line to suspension, that takes a lot of time and effort. The adherent cell lines tend to clump and become, just make aggregates when transitioning too quickly to suspension. So that can create a lot of problems for infectivity. So that's why we have some programs switch to strictly suspension cell lines after adherent, which saves time and can increase productivity, but it also comes with additional costs. So just balancing that for what your program needs. Yeah, so some clients decide to stick with their adherent cell line and gradually wean off the FBS to a suspension cell culture. We've done both, and we support both strategies for development, whatever your program needs.
Okay, thank you, Kelly. So yeah, indeed, this is not just a format change, but something that can really impact the entire process, as you've heard from Kelly. So we have one more questions, actually two. There's two more questions that we can take. So the next one is, what are the main differences between producing first generation adenoviral vectors and helper dependent or gutless vectors? And also, what factors should developers consider when choosing one approach or the other?
So with the first generation ad vectors, you still have the risk of RCA since it has those viral coding regions. And with the HD ad, you don't. So just deciding what you want there. But with the HD ad, you still need to worry about the helper virus contamination since there is helper virus used to co-infect the HD ad, but that can be controlled through process optimization with like co-infection ratios and some downstream process optimization. You also just have to keep in mind that you'll need a specific cell line that produces the enzyme needed to make your HD ad, like Cre recombinase or Cre-Lox P system, for example, which helps reduce helper virus contamination as well. But if you have a larger gene of interest or are concerned about RCA, then HD ad is a great option.
Okay, thank you, Kelly. And that does ties nicely with the last questions that we can take for today, because we have one last question from Michael, and it's around RCA and safety. And the question is, since RCA is an important safety consideration, what process of platform's decision can help reduce RCA risks as program moves from development to manufacturing scale?
So as far as the vector itself, you can choose an HD ad to reduce RCA, or you can choose there are certain cell lines that help reduce RCA, like cap cells. There's some in-process steps you can try to test different MOIs and harvest times post-infection to help RCA. It might reduce your overall yield, though, so just trying to keep that balance and keep that in mind for what's acceptable. We've worked with both HD ad and cap cells programs, so that we're looking to reduce RCA risks, so we support both programs.
Okay, great. Thanks a lot, Kelly. So I guess we are running out of time and we've reached the end of today's session. So I'm sorry if you have not been your question answered, but we'll make sure to follow up via email to have Kelly to answer your questions. Thanks a lot again, Kelly, for sharing your insights, and thank you for everyone that joined the webinar today. As a reminder, the webinar will be available on replay, so you can watch it at your leisure. And of course, feel free, just like Kelly said before, to go visit our website, skfarmteco.com, and also to follow us on LinkedIn to be informed for upcoming webinars, resources, or any company news. Thank you again, everyone, and we hope to see you at future SK Farm Teco webinars.